Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Welcome to the debate. Today we are standing right at the precipice of the bioreactor. You've spent weeks cultivating a high-density fermentation, your cells are packed with product, and now you face the single most critical transition in the entire manufacturing process, the USP-DSP interface.
It really is the moment of truth, isn't it? You're taking this living, breathing, and frankly, often pretty disgusting biological soup, and you're trying to turn it into a clean, processable liquid. We're talking about the engineering of primary cell broth separation.
And this isn't just about plumbing. This one step can account for, 20 to 30% of your total downstream processing costs. It's the first gate. And if you get this wrong, you don't just lose yield. You potentially flood your purification train with impurities that are just impossible to remove later on.
Exactly. And that brings us to the core tension we're debating today. On one side, you have the industrial standard, the, let's call it, brute force of high-speed centrifugation. On the other, you have the more nuanced, mechanism-driven approach of filtration.
So the question is simple. When you're operating at 100,000 liters, is the centrifuge still the king? Or has modern bioprocessing evolved to a point where we should prioritize the gentle touch of filtration, regardless of the price tag? I'm going to argue that for true industrial scale, you need muscle. You need the speed and continuous discharge of centrifugation.
For filtration, specifically depth and microfiltration. My position is that clarity is a false metric if you compromise cell integrity. We need to prioritize Darcy's law over Stokes' law, protecting the product quality from the very first drop.
Okay, let's get into the physics of this, because that is where the battle lines are drawn. I want to start with the reality of the material itself. Microbial fermentation broths are not water. They are high-density, high-viscosity environments. We're routinely seeing solids loading exceeding 10, maybe even 20% weight per volume.
It's essentially a slurry.
Precisely. And when you have that much biomass, you can't simply sieve it out without massive complications. Physics favors density here. That is my primary argument. Centrifugation leverages Stokes' law. We're exploiting the density difference between the cells, usually around 1.05 to 1.1 grams per cubic centimeter, and the aqueous phase, which, sits at 1.
It's a very small window of difference, though.
It is small, but if you apply enough force, it's all you need. At industrial scale, we use disk stack centrifuges generating 5,000 to 8,000 times the force of gravity. That allows us to handle dry cell weights of over 50 grams per liter and, crucially, clear a massive batch in 1 to 2 hours.
See, you focus on the can, but you ignore the should. You mentioned 8,000 times the force of gravity. Think about what that actually means. That isn't just a number on a spec sheet. That is a physical assault on the cell.
We're engineers. We manage energy inputs.
But you're managing biological entities, not mineral ore. My argument for filtration is built on maintaining the harvest state integrity. When you spin cells that hard, especially fragile gram-negative bacteria, you aren't just separating them, you're shredding them.
But let's be real. If the product is intracellular, like an inclusion body in E. coli, we are going to blast those cells open in a homogenizer immediately after this step anyway. Does integrity really matter if the next step is total destruction?
Oh, it absolutely matters. Even if you plan to lyse them later, premature lysis in the centrifuge is uncontrolled. You're releasing a chaotic mix of DNA, lipids, and host cell proteins, HCPs, into the supernatant before you're ready to deal with them. We see data showing that shear exposure in centrifuges can increase the HCP load by 20 to 50% compared to gentler methods. You're just making your own life harder downstream.
That is a fair point on the impurity load, but let's look at the alternative. You champion filtration. You talk about Darcy's law, which governs flow through a porous medium. But Darcy's law has a variable for resistance. And in a high-density broth, that resistance doesn't just go up linearly. It spikes exponentially.
Right. But that's where the technology has evolved. We aren't just pouring broth through a coffee filter here. Whether we're talking about tangential flow, microfiltration, TFMF, or depth filtration, we're using advanced physics. Depth filters, for instance, they utilize a graded porosity matrix.
Graded porosity matrix is a fancy way of saying a thick filter.
It is sophisticated. Large pores catch the big debris, tight pores catch the small stuff. But more importantly, they use adsorptive interactions, electrostatics, and hydrophobic interactions that capture the fines, the colloids, and the lipids that your centrifuge misses completely because they're too light to settle.
But at what cost? And I don't just mean money, I mean operational viability. If I have a fungal fermentation, let's say a filamentous organism that makes the broth act like, non-Newtonian slime, your filter blinds instantly.
Rheology is certainly the enemy of filtration. I grant you that.
Enemy is putting it lightly. If you put a shear-thinning fungal broth into a dead-end filter, the cake resistance builds up so fast your flux hits 0 in minutes. Centrifuges are the only technology that can handle that kind of mud without choking. They can process the unprocessable.
That's true for the most extreme rheologies. But for the vast majority of bacterial and yeast processes, tangential flow microfiltration handles this by using cross-flow velocity. We sweep the membrane surface to keep the cake from building up. It manages the physics.
It manages it until it doesn't. And this brings us to the second major point of contention: economics and operational failure modes. Because even if your filter works, can you afford it?
Let's talk money. I know you view filters as just burning cash.
Because they are, filtration is a massive consumable burden. Depth filters cost anywhere from, $10 to $50 per square meter per use? If you're running a massive campaign, say 200 batches a year, that OpEx is a nightmare. You're buying new filters for every single batch, creating tons of solid waste, and managing a complex supply chain.
It is an operational expense, yes.
Compare that to a centrifuge. It's a steel bowl. It's a fixed asset. You buy it once, you maintain it, and it runs for 20 years. Ideally, it outlasts the molecule itself.
Okay, that is the traditional CapEx versus OpEx argument. But you're glossing over campaign robustness. You say the centrifuge runs for 20 years, but does it run consistently? Large-scale centrifuges, we're talking bowls greater than a meter in diameter. They have scale-dependent failure modes that are terrifying to a plant manager.
Machines break, sure, but we have predictive maintenance.
It's not just breaking, it's the physics of the bowl. Vibration is a constant battle. Seal erosion from abrasive particles leads to leaks. But the big one is the chute.
The discharge.
Exactly. To get the solids out, the bowl has to open a tiny slit while spinning at 5,000 RPM, dump the sludge, and close again in a fraction of a second. If your solids loading fluctuates even slightly, you get torque variations. You can have a chute where the bowl opens and doesn't close properly, dumping your entire product stream into the sludge tank. You can lose a batch in a split second.
That is a dramatic scenario, but accurate. However, filtration has a much more common and insidious failure mode: flux decline.
We call it fouling, but yes.
You call it fouling, I call it the process stopping. Once a filter is fouled by protein adsorption or pore plugging, it is done. You can lose 50 to 80% of your flux. You can't just spin it faster like I can with a centrifuge. You have to stop, clean, or replace. In a continuous manufacturing environment, that downtime is lethal.
Well, actually, modern setups use alternating sanitary replaceable modules. We can switch over without stopping the flow. But I want to pivot back to something you dismissed earlier, the idea that centrifugation handles mud better. There is a hidden failure mode in centrifugation regarding density that you're ignoring.
Go on.
Centrifugation relies entirely on a density difference, Stokes' law. What happens when your cells are fluffy? What happens when you have a filamentous organism that traps water and has a buoyant density almost identical to the broth?
That is difficult.
It's impossible for a centrifuge. Stokes' law has a variable for density difference. If that difference approaches 0, settling velocity approaches 0. It doesn't matter how fast you spin, nothing happens. Filtration separates by size, not density. A filter catches a fluffy cell just as easily as a dense one. That's a fundamental robustness advantage.
I will concede that, for neutral density particles, centrifugation struggles. You can't spin out what won't settle. But there is a low-tech solution we haven't touched on, and it addresses the energy cost issue. Point 3, gravity sedimentation.
Gravity settling in a modern bioprocess? We aren't making wine in a bathtub here.
Hear me out. It is the ultimate low-energy alternative. We are seeing it used effectively in wastewater-adopted systems or organic acid production. Using lamella clarifiers, these are inclined plates that increase the effective settling area. It is simple. It has no moving parts. It uses zero electricity for the separation itself.
It is also excruciatingly slow. We're talking about 4 to 8 hours just to clarify a tank.
For high-value biologics, maybe that's too long. But for industrial enzymes or biocommodities where margins are razor-thin, that low-energy footprint is a massive advantage.
But think about the scale-up physics. In a massive sedimentation tank, you have thermal gradients. If the top of the tank is slightly warmer than the bottom, you get convection currents. Those currents resuspend the solids you just spent 4 hours settling.
You can insulate tanks to manage that.
It's a band-aid. And to make sedimentation work efficiently, you almost always need flocculants. You need to add polymers to glue the cells together into bigger particles so they fall faster.
Flocculants are standard in many heavy industries.
But in GMP bioprocessing, good manufacturing practice, they're a regulatory headache. You have to prove you remove the flocculant. You have to validate the clearance. It adds a chemical impurity to remove a physical impurity. It defeats the purpose of a clean process.
It is niche, I agree. But for robust, flock-forming organisms like yeast, it proves that low tech can work. It challenges the assumption that we always need high-energy inputs.
It challenges it, but I don't think it wins for high-value therapeutics. The risk of contamination and the time cost are just too high.
Centrifuge or filter. But in reality, it's rarely that simple.
Right. We need to synthesize this around the harvest state. You can't pick a separator without knowing the upstream conditions.
Exactly. Late harvest lysis is a killer. If you let the fermentation run too long, cells start dying and breaking apart. This creates fines, tiny cell debris.
And these fines are the worst of both worlds. They blind filters instantly because they plug the pores, but they're also too small to settle in a centrifuge.
This is where the hybrid solution comes in. And I think this is where we might find some agreement. The standard industrial logic is often centrifugation for bulk removal, followed by depth filtration for polishing.
It's the best of both worlds, theoretically. You use the centrifuge to do the heavy lifting, remove 95% of the solids. That protects the filter from being overwhelmed by the sheer mass of the cake.
And the filter protects the downstream from the centrifuge's, let's call it, lack of precision.
Incompetence was the word I was looking for.
Let's stick with lack of precision.
Fair enough. The centrifuge leaves the fines, that sub-micron debris that doesn't settle. If you sent that directly to a chromatography column, you would kill the column. The depth filter acts as the goalkeeper.
I accept that analogy. The depth filter catches the shear-induced debris that my centrifuge might have created.
Precisely. It uses that adsorptive mechanism I mentioned, the electrostatics, the zeta potential, to grab the DNA and the cell wall fragments. So the centrifuge provides the volume handling, and the filtration provides the fidelity.
It handles the volume and the time constraints. By putting the centrifuge first, I reduce the volume that the filter sees, which saves your precious consumable costs. I'm not buying filters to catch 100% of the biomass. I'm buying them to catch the last 5%.
It does extend the life of the filter significantly, but it's worth noting that this hybrid approach requires careful tuning. If the centrifuge shears the cells too much, you generate so many fines that you blind the polishing filter anyway.
That is true. You can't just run the centrifuge at max speed and hope the filter cleans up the mess. You have to tune the G-force to balance clarity with shear. It's a system, not two isolated steps.
And that brings us back to the predictability problem. The hardest part of this entire interface is that you just cannot trust small-scale data.
Absolutely. This is the nightmare of every process engineer. Rheology does not scale linearly. A broth that looks like water in a 1-liter flask might behave like ketchup in a 10,000-liter tank.
And shear thinning behavior is notoriously difficult to model. As you pump it, it gets thinner, but as it sits, it gets thick.
And fouling mechanisms change too. At small scale, you might see pore blocking, just a simple plug. At large scale, with higher pressures and longer runtimes, you see cake compressibility dominance. You get a dense, impermeable layer of cells that you just didn't predict in the lab.
Which means, despite all our theories, despite Stokes and Darcy, the only way to know is pilot-scale testing. Empiricism still beats theory in this field. You have to run the broth to know the broth.
I want to circle back to the time element because I think we move past it too quickly in the rush to discuss hybrid systems. We talked about cost, but we didn't fully explore the cost of time as a quality attribute. In a 100,000-liter fermenter, if I use a slow filtration process, or, heaven forbid, sedimentation, that broth is sitting there for 10, 12, maybe 20 hours.
It's a race against the clock, sure.
It's not just a race, it's a war against entropy. Every minute that broth sits in the harvest tank, the cells are starving, they're stressed. They start releasing proteases, enzymes that chew up proteins. If your product is a protein, your own cells are eating your profit while you wait for the filter to drain. Centrifugation offers continuous, rapid transit. The residence time in the bowl is seconds. The residence time in the harvest loop is minimal. You stop the biological clock.
That is a compelling argument for labile products, certainly. If you have a highly unstable molecule, speed is quality. I won't argue that. But let's look at the flip side of that biological clock. When you run that centrifuge, you're generating heat. We touched on this, but it's critical. The friction of the bowl spinning against the air windage and the liquid acceleration heats the broth.
Cooling jackets are standard.
They are standard, but they aren't perfect. You can easily see a 5 to 10 degrees Celsius rise in temperature during the pass. If your protein aggregates at 30 degrees and your fermenter is at 25, that centrifuge just cooked your product. Filtration, especially depth filtration, is isothermal. There is no heat generation. It's a room temperature process. For a thermosensitive vaccine antigen, that thermal spike from the centrifuge is far more dangerous than an extra 2 hours of processing time.
For muscle. Filtration gives you control. You can control the pressure, you can control the flow rate, and you can control the temperature completely. With a centrifuge, you're strapping yourself to a rocket and hoping the cooling jacket works.
Strapping yourself to a rocket implies a lack of control, which I resent on behalf of centrifuge manufacturers everywhere. Modern machines are incredibly sophisticated. We have variable speed drives, we have automated discharge triggers based on turbidity. We aren't just flying blind.
But you are still limited by the physics of sedimentation in a centrifugal field. Let's dig deeper into the fines issue, because this is where the economic argument for filtration actually gets stronger, counterintuitively. You said filters are expensive.
They are.
But what's the cost of a failed chromatography column? If your centrifuge lets through 5% of the solids, mostly lipids and cell debris, that material hits your protein A column or your ion exchange resin. It binds irreversibly, it fouls the expensive resin beads.
That is why we have guard columns.
Which are another cost. But if you use a high-performance depth filter, you're removing those lipids and colloids before they ever see the resin. You might spend $50,000 on filters for a campaign, but if you extend the life of your $1 million resin pack by 20%, you've saved money. The filter sacrifices itself to save the chromatography. The centrifuge doesn't make that sacrifice. It just passes the problem downstream.
That is a valid holistic economic view. I often look at the unit operation in isolation, separation cost per liter. You're looking at the total process cost.
You have to. Bioprocessing is an interconnected ecosystem. You can't optimize one step at the expense of the next.
I will give you that. However, I want to challenge you on the scalability of that holistic view. It works beautifully for high-value antibodies or vaccines, where the product is worth thousands of dollars per gram. But what about the bulk stuff? Industrial enzymes, bio-based chemicals.
The margin game changes, obviously.
It changes completely. If I'm selling an enzyme for laundry detergent, I cannot afford depth filters, period. The economics don't close. I need a decanter centrifuge or nozzle separator. I need to process 500 cubic meters a day. The impurity load you're so worried about, it doesn't matter as much. We aren't injecting this into humans.
In the commodity space, yes. Volume is king. And I will concede that filtration simply doesn't scale to the white biotechnology levels. Biofuels, food ingredients, the surface area requirements would be the size of a football field.
Exactly. So, we have a divergence in the industry. Pharma and high-value biologics can afford the luxury of filtration's fidelity. Industrial bio needs the efficiency of centrifugation.
I wouldn't call it a luxury, I'd call it a regulatory necessity. The FDA or EMA expect a certain level of purity and process control. If your centrifuge varies in performance because of a slight change in broth density, that's a deviation. A filter is binary. It either holds the particle or it plugs. It rarely leaks.
It rarely leaks, but it can rupture. If you overpressurize a depth filter housing, you can bypass the media.
True, but pressure sensors make that easy to detect. A leaky centrifuge is much harder to spot in real time until you see the turbidity spike in the harvest tank.
We're getting into the weeds of failure modes again, but I think it highlights the complexity. Let's talk about the future for a moment. We've discussed the current state, centrifugation, filtration, the occasional sedimentation, and the hybrid approach. Where is this going?
I think we're seeing a move towards single-use centrifugation.
Interesting. Getting rid of the steel bowl?
For smaller scales, yes. The cleaning validation, sip and sip, for a large steel centrifuge is a nightmare. It takes hours, uses tons of caustic and water. Single-use centrifuges, plastic inserts that you spin and toss, are entering the market.
They are, but they are currently limited in G-force and capacity. They can't match the 8,000 Gs of a disk stack.
Not yet. But they bridge the gap. They offer the speed of separation without the cleaning downtime. It's an attempt to get the muscle without the maintenance.
On the filtration side, I see advancements in materials. New membranes that are more resistant to fouling.
Yes, and better depth media. We're seeing charged depth filters that are specifically tuned to remove DNA or specific host cell proteins. It's becoming a chromatography step disguised as a filtration step.
That blurs the line between USP and DSP even further.
Which is good. The interface should be a gradient, not a wall.
I agree. But for now, for the listeners running a plant today, we have to deal with the tools we have. So as we wrap up, let me summarize my position. I look at the manufacturing scale 10,000 liters and up, and I see a volume problem. You need throughput. You need to beat the clock to prevent degradation. Centrifugation is the only technology that offers the brute force capacity to handle high solids microbial broths efficiently. It's a mature, GMP-validated workhorse. It has its flaws with shear, yes, but those are manageable risks compared to the operational nightmare of changing thousands of filters.
And my position remains that this is fundamentally a quality problem. I argue that filtration provides the necessary finesse. It protects the product quality and, crucially, protects the downstream equipment from the invisible burden of fine debris and impurities. While the centrifuge is fast, it's messy. Filtration, especially depth filtration, uses mechanism, not just force, to ensure that what goes downstream is actually processable. It is worth the cost because it ensures the integrity of the molecule.
So we leave you, the listener, with the reality of the bioprocess engineer. There is no single best technology. There's only the technology that fits your specific broth rheology and your product's sensitivity.
It is a choice between speed and integrity, muscle and mechanism. And usually the answer lies somewhere in that messy middle.
Thank you for joining the debate.
Until next time.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.